We are starting. We will be learning about anaerobic organisms today. Anaerobic organisms are different than the usual common aerobic organisms as we know. What are the common features of anaerobic infections are? Many of them are derived endogenically, organisms which are already present in the body. But when anaerobic conditions get created in the body like trauma, necrosis, foreign body impactment, in such conditions, these endogenous organisms can create anaerobic infections. Most of the anaerobic infections are polymicrobial, multiple bacteria involved in causing infection, and they produce a very putrid, foul smelling odor along with cellulitis. In anaerobic organisms, we have a wide spectrum of organisms in which we can have obligate or strict anaerobes which cannot withstand small amount of oxygen also, and we have some facultative anaerobes which can withstand and grow in presence of small amount of oxygen. Then we have another two groups, that is spore forming anaerobes and non-spore forming anaerobes. Main pathogens in spore forming anaerobes are Clostridia, and the three main groups of Clostridia which can cause human infections are: one, the Clostridia which can cause gas gangrene, that is perfringens or Welshi; then organisms causing tetanus, Clostridium tetani; and Clostridia causing botulism are Clostridium botulinum organisms. In non-sporing anaerobes, we have multiple organisms. We have cocci like Peptostreptococci. Then mainly we have gram-positive bacilli like Propionibacterium, Lactobacilli. In gram-negative bacilli, commonly which cause anaerobic infections are Bacteroides, Porphyromonas or Prevotella. These are the commonly seen gram-negative non-sporing anaerobes. What are the common infections caused by non-sporing anaerobes? They can cause brain abscesses. In oral cavity, they can cause Vincent's angina, which is cellulitis kind of oral cavity, subcutaneous infection or submucosal infection. They can also cause ulcerative gingivitis, dental abscesses in multiple organs. Another common infection is aspiration pneumonia by non-sporing anaerobes. This is a picture you can see of gingivitis, kind of periodontitis caused by, it's a polymicrobial infection again by non-sporing anaerobes and other organisms. Whitish patch you can say, Vincent's angina, sometimes can present as a severe oral cavity infection. This is morphologically how different anaerobes appear, and you have spores. So they can have either bulging spores or non-bulging spores. No. Bulging spores are where the spore doesn't bulge the body of the vegetative bacteria, and in bulging we have like a round terminal spores we have for Clostridium tetani, or subterminal oval spores as we have in Clostridium welshi. So we'll see different Clostridia that can cause human infection. Clostridium welshi or perfringens, both names are used for this organism. They are thick gram-positive bacilli with truncated ends. They can be pleomorphic in appearance. They rarely produce spores in human body, but they produce spores in external environment, in cultures also. Many strains have ability to grow at higher temperatures, 45 degrees or so, and they can grow faster at this, and this is used for laboratory diagnosis. They produce a lot of virulence factors, multiple toxins and enzymes they can produce. They can invade tissue, and they can cause cellulitis, along with they can invade muscle tissue and bloodstream also. So they can also cause septicemia and bacteremia. And main prominent toxin producer alpha, beta, epsilon and iota. These are the toxins, and a severe infection. That's why, because they are prolific, you can say, as far as pathogenicity is concerned and toxin production is concerned. Disease caused is mainly gas gangrene, which is also kind of clostridial myonecrosis, and so there is initially edema. Is a lot of edema along with myonecrosis in wounds, especially seen in wounds where there is severe muscle crushing. So roadside accidents in wars, crushing is there and contamination is there. So anaerobic conditions are created in such conditions and infection starts from here. Again, this is also polymicrobial infection, and along with predominantly perfringens, along with other organisms are involved in this. These are pictures of gas gangrene. As you can see, severe necrosis is there. These are some of the tests used for lab diagnosis of Clostridium welchi. You grow the organism and perform Naegler's reaction or Lacey-Perrins test to confirm welchi. Then we have this reverse cam test can be done to confirm the growth of Clostridium welchi. This is litmus, stormy fermentation of litmus milk for confirmation of Clostridium welchi. Then we have tetanus, another organism, Clostridium tetani, which causes tetanus. And these are slender gram-positive bacilli with terminal spores, round spores, unique appearance. They are obligate anaerobes with strict anaerobes, but they do not invade, and so they remain localized at the site wherever they get put in the body, and they produce exotoxins, and the effect is because of the exotoxin which spreads in the body. Then easily growable organisms, and they produce mainly two toxins, titanolysin and titanospasmin. The main effect of the infection is brought about by titanospasmin. What this titanospasmin does is that it travels through nerves and it goes, the main action is in spinal cord. It inhibits the balancing effect that is seen in the spinal cord. Spinal cord has inhibition for so that our both flexors and extensors do not act at the same time, but this inhibition is removed, and so there is simultaneous contraction of agonist and antagonist, leading to rigidity and spasm in the body. That's the effect of the titanospasmin. Tetanus follows due to a penetrating injury. Anaerobic conditions are created in there, and the toxin produce, and then the effect, which leads to the presentation of tetanus as such. Usually, the diagnosis is by clinical presentation because the organism you may not be able to grow the organism, and of course, growing the organism takes more time. So the treatment or whatever measures you have to be done immediately. So laboratory diagnosis has a limited scope in case of diagnosis of tetanus as such. But you have a lot of preventive measures are available to prevent active immunization is available using tetanus toxide, formalin activated. Main vaccination consists of 3 doses, and booster doses can be given after many years if required. Passive immunization is available for immediate protection. In case a person is not vaccinated, you can use passive immunization for immediate protection against the disease as such. And the last organism is Clostridium botulinum, which causes botulism. It's kind of a food poisoning, gram-positive bacilli, spore-bearing. They get ingested, and a powerful exotoxin they produce in GI tract, which gets absorbed, and then it has the effect on nerves. It blocks the release of acetylcholine at synapses, and it leads to paralysis is the overall effect in the body, dysphagia. Ultimately, it may be severe and may cause respiratory muscle paralysis, and death may follow because of that. So, it's usually produced by consumption of preserved or canned food or improperly cooked food. Thank you.